I opted to utilize our general biology sequence and specifically our ecology unit for my research project. A couple of reasons why I picked the general biology sequence. It's really that first introduction to a college level science course that a lot of our students at Ellsworth experience. And so it's that first impression of what science is really like at the college level and the rigor of science. And because of that, we often have a really high drop rate for general biology. We encourage our advisors to put biology majors into general biology rather than non-majors, which have other options like intro and chemistry and so on, but sometimes that doesn't happen. And so we do have a really high drop rate. For example, the first year I taught general biology at Ellsworth. We had 3 sections of intro or of general biology 1 in the fall, and we lost about a third of the students when we moved into general biology 2 in the spring. And so we see that continuously in this class with lots of students dropping or just kind of giving up on the class. And so I wanted to try to incorporate more research-based studies into the biology course so that students would have, that feeling of belonging, right? Something that they own, that they can be working on throughout the semester, and that will keep them involved and encourage them to even make it through some of that difficult stuff so they can keep working on this project that they developed on their own. So to incorporate that water aspect to our project, I found that the ecology unit worked best for that. And so I was able to incorporate this research project into Gen Bio 1, utilizing these 2 learning outcomes and kind of making this the assessment project for these learning outcomes. So our ecology learning outcome, and then just also understanding the process of science and how evidence really plays a role in developing scientific conclusions. So as part of this project, I wanted to build those partnerships. We already had a pretty good partnership with Hardin County Conservation and Calkins Nature Area. I'm also, in addition to teaching the general biology courses, I was the head of the conservation technology program. And so we worked very closely with Hardin County Conservation, and we used the Calkins Nature Area as one of our outdoor classrooms for that program. But I wanted to expose my non-conservation students to this amazing resource, these awesome walking trails. You've got pretty much every different type of habitat found in Iowa located at Calkins Nature Area. So it's a great place for students to explore and get out and just enjoy the outdoors. So if you've never been to Calkins Nature Area, it's about a 76 acre piece of land that borders the Iowa River. So here's the Iowa River up here in the northern boundary of the park. And then there's a bunch of trails that run through it. But the part that I found most fascinating for this project is there's also a drainage creek that runs through Calkins Nature Area. And so this drainage creek, it's hard to tell with this picture, but there is farmland surrounding all of Calkins Nature Area on all sides except for where the Iowa River is. And so this drainage creek is actually a tile drainage creek. And so that farmland has tile that eventually drains into this creek, and then that creek drains into the Iowa River. Quite a few years ago, Hardin County took on an initiative to help improve the water quality of the Iowa River. And so they built a man-made wetland into the drainage creek on the property of Calkins Nature Area with the idea that this wetland would help filter out some of those impurities before that water actually entered the Iowa River. And so you can see the drainage creek running through Calkins Nature Area and entering the Iowa River, and this large portion right here is obviously that wetland that they built. They often go out with classes and students and do sampling just randomly, but they've never actually kept a data set. So they don't really have an idea of how well the wetland is really working in that area. And so the collaboration with Ellsworth then was to start a long-term data set where we would be able to collect water quality data and then also macroinvertebrate biodiversity data. And so some of the water quality, we ran the whole gambit. We collect nitrogen, phosphates, pH, turbidity, oxygen concentrations, and then also BOD. So we looked at oxygen concentrations after a week. And then we also collected macroinvertebrates using kick nets. And we were able to identify those species to the family level. The original plan was to use barcoding as part of the project to be able to identify specific at the species level, but we never got that far with the project. Hopefully, maybe in the future they can do something with that. And so students would be exposed to this project, and we would collect samples above the wetland and below the wetland. So students would have an idea of how the wetland was actually affecting the water quality. And if they were really into the project and were really interested in it, they could also collect samples within the wetland to get that as well. So the first time I ran this project would have been in the fall of 2019 in General Biology 1. At that point, we were teaching our ecology unit in Gen Bio 1, and it worked really well in the fall. We have pretty nice weather up through the end of October, and so it gave students plenty of time to get out into the field and do stuff. And so I started this project kind of as a whole semester project for Gen Bio 1. In September, the ecology unit was that first unit that we went over in lecture. And then in the lab portion, I started building that foundation with my students. We did a scientific methods lab. Then we had a day in the library where students did research. So they were introduced to all of these different water quality parameters and macroinvertebrates. And then they were told to pick one of those parameters that they wanted to focus on for their project. And they were told that they also had to do macroinvertebrates. So they had to do the macroinvertebrate survey, and then they were going to focus in on their research for one of those water quality parameters. And so they spent that day in the library doing research. And then by the following week, as a group of usually 3 to 4 students, they would have to have submitted their research question and their hypothesis, which I developed, or all students had to have their hypothesis in what we called the if and then format. And so that was my opportunity to make sure the students really understood what they were doing, what their variables were, and I could provide feedback at that point before we actually went out into the field and started doing anything. Then in October, we spent more time with that foundation sort of thing where we analyzed a research paper as one of our labs. I also spent some time that day talking to students about why it's important that they understand how to read a research paper, the different parts of the paper and how they're connected, and how they can use that same format of a research paper for their final project. So all of their lab reports were designed in that same format as a research paper. And then I feel very strongly about incorporating statistics into my biology courses. And so we spent a day doing statistics review. So students learn the basics about chi-squares, t-tests, ANOVAs, and regressions. They were very briefly introduced how to do some of that stuff in Excel and provided with kind of outlines on how to run those different tests in Excel and when those different tests should be used. And then really we spent a lot of time trying to make sure that they understood the output of those tests, right? So what is a p-value? How can you take that information and relate it back to your hypothesis and draw conclusions about your results? Then finally, they were able to go out and do a field day where they collected all of their data. This is a picture of the wetland out at Calkins. So the creek comes in on the left-hand side, moves through the wetland, and then continues on the right-hand side through the rest of the creek and out to the Iowa River. So students, this was probably their favorite day of the whole semester. They were able to put waders on and get in the water and have a lot of fun using the kick nets and the turbidity tubes and collecting those water samples. In the field, I had all students collect samples so that they would in class have to analyze all the different water quality parameters, even though their project was only going to be on one of the parameters. And this was so that students would have a larger data set to use, right? So they could use everybody's data from the class instead of just a single sample that they collected. So then the following week, we actually, in lab, we did the water quality analysis. We used titrations for most of the water quality analysis. And then we also did macroinvertebrate identification in lab that week. And then in November, the first draft of their lab reports were due. The first draft was primarily the introduction, which included moving from kind of that broad information down to the purpose of their paper, and finally the hypothesis. I really focused a lot on the structure of a research paper for this class, and so it was really important that they followed that format. And then also writing out the results, or not the results section, but the method section of their project. Then the second draft was kind of more like a final draft where everything was incorporated. They received feedback from a peer and then also feedback from me. And if they felt like they needed to make changes for the final draft, they could. If not, I left it up to them with the understanding that my suggestions were to help improve their paper, but their paper is their project. And so it would be up to them on how they wanted it graded. So as I said, I used this project to assess some of the learning outcomes. And so a standardized rubric was used to grade their lab reports or their research papers. And so that rubric included a section for the introduction, the development of a question, hypothesis, variables, and so on. And you can see that for the most part, students did relatively well with these first things where they actually had that first draft and then the second draft. And so they had multiple opportunities to gain feedback on those areas. But a lot of students struggled then with the actual experimental design portion. I think a lot of that was just communication between me and the students. You know, I provided them with the rubrics, but we didn't go over it in class. And so I think a lot of students didn't really pay attention to the fact that they had everything they needed right in front of them. And so that was one of the things that I knew I would need to change, is spending more time going over everything with them multiple times. Something else I noticed then was that in the experimental design, students really struggled connecting their method section to their actual question and hypothesis. So as I said, students collected data and actually did water quality analysis on all of the parameters, even though their project was only over one parameter. And so in their method section of their lab report, they included information about all of those parameters instead of just the one that their project was over. And so it was really, I needed to explain to them the purpose of the method section and the purpose of that part of a research paper for communicating the results. And then for some reason, even though we spent some time identifying variables and it was clearly labeled as a part of the rubric, the students needed to clearly ID their controls, their independent and dependent variables. Students just didn't do that in a clear way in their methods section. Students also struggled with communicating the results. So during the first draft, I noticed that students didn't understand the difference between raw data and summary data. And so students would give me a graph that had a bar for each piece of raw data instead of just a bar with the averages. But finally, by the end of the project, students were able to get that down in their final lab report. But then a lot of students were trying to interpret their results in the results section, which was something that we also went over, in that the results section is just for providing the information. The conclusion and discussion is where you actually interpret the results. And a lot of this might just have been, I had really high standards for my Gen Bio students, and maybe they were too high of standards for first time experiences with research. And then in the discussions, they also struggled with then trying to bring that back to the big picture. Why are we doing this? What's the purpose? What sort of data is already out there that you can use to support your results, or how can your results help somebody else? And so those were just things that I noticed that students needed to work on. And so what this meant for me is that I really wanted to incorporate more research papers and analyzing papers into my project. I really wanted to allow students to examine other lab reports and do more peer review of lab reports, so that they could get an idea of what's a good lab report, what's not, how to critique them, and so on. And then also statistics. Again, if I'm going to make students actually do statistical analysis for this, which I did, it means I'm going to have to spend more time teaching them about the statistics, right? So that might include an introduction to statistics and how to do it in Excel, doing some different statistical output analysis activities, so that, you know, if you have this p-value, what does this mean for this hypothesis, and so on. And then also just there are other lab reports that they do throughout the semester, and so being able to incorporate statistics into those lab reports too, so they have more practice, I think is going to be important. As I said, retention was a big issue for Gen Bio 1, and so I wanted to look a little bit at the data for retention as well. And what I saw was that, you know, for the most part, grades actually improved by almost 5 percentage points between fall 2018 and fall 2019 when I completed the project. And so I think students were just more in tune with the class and more willing to continue working through some of that harder stuff because they had this project that they were working on. I also saw with drop rates that, yes, we had quite a few drops in August that first week, but that's pretty normal when students realize, you know, maybe this isn't the class for them. But then in compared to 2018 and 19, I had relatively few drops after we actually started the project, which I saw as very promising, showing that this project really did help keep the attention of students. So then very quickly, Ellsworth and Marshalltown both went through a flip-flop of learning outcomes. So what was taught in Bio 1 is now taught in Bio 2. And so that completely changed the whole outline of my project. And so I was actually able to do this project again in Biology 2 during this past year with COVID-19. And so I took advantage of the fact that we were in that A/B model for labs as well, where students would do an online lab one week and then come to class the next week and then flip-flop in the fall. And because of that, I was able to do a lot of groundwork and really kind of revamp my lab curriculum to make it all about the scientific process, right? And so students did a scientific methods lab. They did an introduction to statistics lab. They did a whole Excel tutorial through HHMI. They have an awesome Excel tutorial lab that students can complete. Then they did their first lab report with an experimental design lab. Then they analyzed a couple of papers in Bio 1, and they actually did a final project in Bio 1 too, just to keep them focused and in this idea that they own something in this class. And so their final lab report for Bio 1 was a photosynthesis lab, where they were able to manipulate different variables how they wanted to. And then in the spring, this last couple of months, I actually completed the water quality lab again. And so in the spring, again, we started out by talking about experimental design and reviewing statistics. They actually did 3 lab reports in the spring. The first was on a genetics lab, which everybody's was the same, but it helped them get back into that mind frame of writing lab reports and research papers. They analyzed another research paper, and for each one of those, I have like a worksheet that they follow along with to help them get the most out of it. And then at the end of March, we actually were finally able to get to our ecology unit. And so we did a water toxicity lab where students worked with water daphnia and looked at different toxins and how it affected their heart rate. So that was their second lab report. And that was also their introduction to water quality. And then we did a research day where students again had to develop, they were introduced to all these different parameters. They did research in lab, they developed a question and hypothesis and identified their variables. We went out into the field again. And then the next week we did water analysis and macroinvertebrate identification. And then another week we spent talking specifically about statistics for this project. And it was kind of like a research work day for them to be able to ask any questions that they had in lab. There were other labs thrown in here. Obviously, this isn't 16 weeks worth of labs, but these were just the labs that were dedicated specifically to this project. So here's a comparison of the learning outcomes for fall in Gen Bio 1 versus spring Gen Bio 2 when I did the project. And I see, they did basically pretty similar for the intro question and hypothesis, but where students struggled in Gen Bio 1, they did a lot better in Gen Bio 2 with the experimental design and results section, which makes sense because they had basically 2 whole semesters of foundational work to build up to this project. In Gen Bio 2, we don't really see a lot of drops. So there were no drops in spring 2019 versus spring 2021. So I went way back to before COVID hit for this data just to have it kind of more even comparison because last spring was chaotic for everybody. And so I didn't really see that big of a difference in drop rates. So like I said, we didn't have a lot of drops. There wasn't that big of a difference in grades, overall grades either, by doing this project in the spring. It looks like there's a large difference here, but there's not. It's less than a percentage point in grade difference between spring 2019 and 2021. So final thoughts. I felt like incorporating the research project and making those changes to Bio 1 had a larger impact on overall retention and grades. Students did complete a research project in fall 2020 with that photosynthesis lab. And so they were still, even though they didn't do the water quality lab, they were still owning a project at the end of the semester. And so I think that continued to help boost grades in that semester. And even though I didn't see a change in overall grades, I did see a boost in student morale. So I'm not sure about all of the other community colleges, but we did not have a spring break this past spring, and it really, really affected our students mentally and physically. They were just done by the end of the semester. And having this project to look forward to and being outside and doing something that's different and not that monotonous lab stuff or lecture stuff really helped them, I think, finish out the spring semester as strong as they could. And because ecology will remain in the second semester of this sequence, I think it's going to be important that if I'm going to make this a full project for Bio 2, that the project needs to be introduced at the beginning of the semester, and students can spend more time maybe on that research aspect of the project and gathering that information and doing sort of like a lit review to make it more of a semester-long capstone project. And then I will always say this, that I really think it's important that we incorporate at least an introduction to statistics in our biology sequence. From working with my students, they would see, I'm going to move back here for a minute, they would see this graph and say that there's a significant difference between the grades, the average grades in spring 19 and spring 2021. And there's not. If I actually did a t-test on this, there's not a significant difference. And so, but they see that difference in the bars, and so they automatically assume that that means that there was a significant difference in the results. And so explaining to them the importance of a p-value and what it means and how scientists use that to communicate their results, I think is really important. And then the last thing that I learned about this project, it's not about adding a single research aspect, at least not for me, it wasn't, but rather about really overhauling the entire lab curriculum for the biology sequence and making it very much more scientific and critical thinking based throughout the entire semester, instead of those cookie cutter labs that I started with, 4 or 5 years ago. So, and these are pictures of my students doing some of their research. So any questions? I'm sorry I went over.
No, that's great. Thank you for sharing. And by the way, for everybody, if you're willing to share your PowerPoints, if you had one with me, that'd be great as well for the website eventually. Do you think you're going to go back to this area then and continue testing, or?
So, unfortunately, fortunately, I guess it depends on your point of view. I've actually taken a position at Henry Ford College in Michigan, and so I won't be back at Ellsworth next year. I know. And I decided to move closer to family. I'm from Michigan, Indiana area, and so it was just kind of the right move for us at the time. But I hope that the person that replaces me will continue to have that strong connection with Hardin County Conservation. And I plan on working with the person that replaces me if they want my help, obviously, and providing them with this research project to continue collecting that data. We did run into a couple of big bumps with this data. The first year that we did it in the fall, we had a ridiculous amount of rain that October. And so it was interesting to see students' analysis of the results and explanation for why they got the results they did, trying to bring it back to the changes in the weather. And then this spring, we were in a drought. And so we had the opposite problem where the wetland wasn't moving in. The creek bed was completely dry above the wetland and barely any water below the wetland. And so again, looking at how students interpreted the results in comparison to the movement of the water was pretty interesting. So I hope that they continue collecting data from this area.
I have a quick question.
Sure.
Jessica, do you have other resources you'd be willing to share? I agree with your comment wholeheartedly about Excel and statistics. I've used the HHMI thing before, but do you have other resources that you might share?
Sure. I actually put together, because my students were doing the half online, I put together an online PowerPoint presentation recorded that goes through all of those different statistical things and incorporates the Excel worksheets. And so it's kind of like a multi-dimensional lab. I'd be happy to share that information with you guys. Yeah.
That would be great. Thank you so much.
Yep.